Welcome back, friends. We in the last video we discussed about some MCQs on seed dormancy, and in this video, in continuation with that, we'll have more discussion on the next phenomenon, that is seed germination, from your plant physiology paper. Okay, so again, this is a pea plant showing seed germination, and this is a typical example of hypogeal or hypogeous germination, P. What is that we are going to discuss? Okay, so the very first question, when the hypocotyl, you have studied the embryo axis has a radical and plumule, and the stem axis or the internode between radical and the cotyledon, which are present usually at the center. So radical and cotyledon, it is called as hypocotyl, whereas the internode between cotyledon and the plumule, or cotyledon and the first true leaves that are formed later, that is called as epicotyl. So when the hypocotyl and hence the cotyledons come above the soil first, the seed germination is called as. So this type of seed germination in which cotyledons are above the soil is called as epigeal. Epi means above, geal means soil. Above the soil, above, geal means earth or soil. Above the soil, epigeal or epigeous. Who is above the soil? Cotyledons. Cotyledons are above the soil. And who has brought them above the soil? It is the hypocotyl. It forms a hook-like structure, and it is called as hypocotyl hook. So that hypocotyl hook actually pulls up the cotyledons above the soil. So this method of seed germination is called as epigeal or epigeous seed germination. So answer is option A. Second question, when the epicotyl comes above the soil, that means hypocotyl remains below, and therefore cotyledons remains below the soil, then the seed germination is called as, so cotyledons soil below the soil, so hypogeal, it is called as hypogeal. Hypo means below, geal means soil, hypogeal or hypogeous. Who is below the soil? Cotyledons are below the soil during seed germination. So this is hypogeal type of seed germination. So answer here is option B. In which of the following seeds seed germination is epigeal or epigeous? So options are given. Now peas and maize and coconut, these are the examples of hypogeal. So you must know the examples. So here epigeal or epigeous type of seed germination is shown by beans. So here answer is B. In viviparous type of seed germination, another type of seed germination, apart from epigeal seed germination and hypogeal seed germination, there is one more germination type that is called as viviparous seed germination, or phenomenon is called as vivipary. Here in this seed germination, the seeds undergo. So this question has been asked in civil services examination, preliminary examination 2010. Nowadays they don't take for preliminary exam, they don't take objective optional paper, but some time before they used to ask such type of questions, objective questions. So here in viviparous type of seed germination, the seeds undergo 3 months of resting period, 6 months of resting period, 12 months of resting period, and no resting period. Now, you know, viviparous germination is an extreme stage of seed germination or extreme seed dormancy, in which there is at all no seed dormancy. Seeds germinate immediately, even when they are attached to the parent plant. Even when still they are attached to the mother plant and they start germination. So that germination is called as vivipary or viviparous type of germination, which is exhibited, which is shown by some species of mangroves, the plants which are growing in some saline conditions, waterlogged areas, your Sundarban regions. So such a plants show viviparous type of germination, in which there is no resting period, there is no dormancy at all. So here answer is option D. Which of the following is the major storage tissue? The reserve food material is stored in the seeds that provides food to the developing embryo during germination in albuminous seeds. Albuminous seeds means endospermic seeds. Non-endospermic seeds are called as ex-albuminous. Ex means the endosperm was present previously in the seed, but now it is not there. It is absent in the seed. But this doesn't mean that the food material is not present in the seed. Whatever endosperm that was present in the seed previously, it was being used up. It was utilized by the embryo and it was stored now in the cotyledons of embryo. So food material in endospermic seed is stored in the endosperm. So they are called as albuminous seeds. Albumin means endosperm. And in case of non-endospermic seed, food material is there, but not in the endosperm. It is present in the cotyledons. And such a seeds are called as ex-albuminous seeds. So here the question is, where is the site of major storage tissue? Which tissue provides food material to the developing embryo during germination in albuminous seeds? That means in case of endospermic seeds. So obviously, if you see the options, obviously the answer is endosperm. This is the major storage tissue. Endosperm is the major storage tissue that provides food to the developing embryo during seed germination in albuminous seeds or in endospermic seeds. A, endosperm is the answer. Same question, which of the following is the major storage tissue that provides food to the developing embryo during germination of ex-albuminous seeds, that is non-endospermic seeds? So obviously this is not the answer. So here answer is cotyledons, but because the food material is stored in the cotyledons of embryo. So B is the answer. So matching type of question, name of the plant is given in the first list, and in the second list types of seeds are given, and you have to match with the help of code given A, B, C, D. So you can see a dicot, monocots, you know, pea, beans, castor, these are dicots. Orchids and cereals, these are monocots. So you have to identify which are the albuminous seeds and which are ex-albuminous seeds, which are endospermic seeds and which are not endospermic seeds. So pea, beans and gram, these are dicots with ex-albuminous, because in case of pea, beans, gram, the endosperm is absent and whatever food material is there, it is stored in cotyledons. So A matches with 3. In case of castor and poppy, these are dicots, but these are albuminous, endospermic. Most of the seed, most of the oil seed plants, oily seeds, they are endospermic, albuminous seeds. So B matches with the first. Orchids are the examples of monocots which are ex-albuminous, and cereals and coconuts, wheat, rice, jowar, your bajra, maize, all these are monocots with albuminous because they consist of albumin or they consist of endosperm. Their endosperm is starchy endosperm. That is the edible part of cereal seeds. So after matching, you will get the answer. Answer option A is the correct answer. Again, matching type question, and this question appeared in civil services prelim exam of 2010. Name of the plant was given and types of seeds to which the given plants belong was given, and you have to match similarly. So again, you can match. Mango is dicot and it is ex-albuminous type of seed because it has no endosperm. It has cotyledons as a reserve food material storage tissue. Castor again, it is dicot albuminous, and orchid again monocot ex-albuminous, and barley, which is an example of cereal, it is monocot and albuminous. So again, after matching, we will get the correct answer, and that is option A. Which of the following is the major storage tissue that provides food to the developing embryo during germination in orchid seeds, monocot seeds? So whether they are whether the storage tissue is endosperm or perisperm or cotyledon or what, you know, orchid seeds are example of monocot seeds which are ex-albuminous. But here none of these is the answer because these are very small and there is no endosperm, there is no cotyledon. So D is the answer. And you can see here in nature, the orchid seeds germinate. The word should be germinate here. Orchid seeds germinate only in association with. Again, civil services exam question. Okay, so you should know this, that orchid seeds germinate only in association with mycorrhiza, because there is no other major storage tissue like endosperm or your cotyledons. So orchid seeds depend on mycorrhizal association, symbiotic association for germination. Orchid seeds germinate only in presence of mycorrhiza. B is the answer. The enzymes like alpha amylase that degrade the storage materials, that is starch, are synthesized in which part of monocot seeds? So we have studied the structure of monocot seed. There is embryo with radical, plumule, radical with a coleorhiza, plumule with coleoptile, and there is a single cotyledon which is called a scutellum, and there is endosperm, and this endosperm is surrounded by a proteinaceous layer, living layer, and that is called as alluran layer. And the enzymes alpha amylase are synthesized in this alluran layer. Okay, because so answer is B, because you studied that the scutellum and the embryo axis, the part of embryo, they produce the secrete enzyme, that enzyme or hormone, properly hormone, gibberellic acid, and that gibberellic acid initiates formation of hydrolytic enzymes in the alluran layer, and these hydrolytic enzymes then are responsible for the mobilization of storage material. So here answer is B. Okay, the carbohydrates are stored in seeds, mostly in the form of, they are stored mostly in the form of starch, and they are then degraded with the help of enzyme alpha amylase and beta amylase, and simple sugars are formed. So C is the answer. The lipids are stored in seeds in the form of, so lipids are stored in the form of triacylglycerides, and these triacylglycerides are degraded with the help of hydrolytic enzyme known as lipase into fatty acid and glycerol, a type of alcohol. Okay, and that fatty acid then is transported to glyoxysome, where it is converted into organic acids, malic acid, and like that. So that you have studied, glyoxysomes are present there, and then these organic acids then get ultimately converted into sugar, and that process is called as gluconeogenesis. That is the synthesis of glucose from non-glucose material. So that you have studied. So here answer is B, triacylglycerides. Which of the following organelles are present only in oil storing seeds? So this is a very interesting question. Spirosomes, peroxisomes, glyoxysomes, ribosomes. So here answer is glyoxysomes. Glyoxysome and glyoxylate cycle is present. It is unique only to oil storing seeds which are germinating. Germinating oil storing seeds show presence of glyoxysomes and glyoxylate cycle. So C is the answer. Which of the following enzymes stimulate germination in barley seeds? Barley seeds are the examples of cereal seeds, monocot seeds, cereals. And you know cereals, seeds of cereals, wheat, rice, jowar, bajra, maize, barley, they are having endosperm. They are albuminous seeds. They have starchy endosperm, and that starchy endosperm, which consists of starch, is acted upon by enzyme alpha amylase and forms simple sugars. So here, as the question is regarding barley seeds, answer is alpha amylase. If the question were regarding oily seeds, then the answer would have been lipase. So here answer is alpha amylase. C. Which of the following hormones interact antagonistically, opposite with each other? They are having opposite action, exactly opposite of each other with respect to the seed dormancy and seed germination. Auxins, gibberellins, gibberellin cytokinins, gibberellins abscisic acid, auxins and abscisic acids. So which is that pair which works antagonistically? And you know, you have studied, this is the pair gibberellins and abscisic acid. You know that gibberellins are responsible for seed germination. Abscisic acid is responsible for seed dormancy. And seed dormancy and seed germination are exactly opposite of each other. So these hormones are oppositely working exactly opposite, that is antagonistic. So here the answer is gibberellins. C is the answer. Sorry, gibberellins and abscisic acid. Option C. Then the role of gibberellins, GA3, mostly in seed germination is what it does. It facilitates oxygen intake or increase incubation rate or activate or synthesize hydrolytic enzymes. All of this. What is the role of gibberellins? We know the embryo of the seed during seed germination produces gibberellins, and these gibberellins go to aileron layer, and it initiates activation of the enzymes, hydrolytic enzymes, which are already present there, or it also initiates the synthesis of new hydrolytic enzymes, which will be required to mobilize the storage reserve food material. So here the answer is C. It activates or synthesizes hydrolytic enzymes like your protease, peptidase, lipase, then what is left? Alpha amylase, like that. So answer option C. C is the answer. Okay, matching type question again. Name of the plants are given in the first list, and the second list type of seed to which they belong. Now we have studied different types of seeds: albuminous seeds, ex-albuminous seeds, that is endospermic seed and non-endospermic seed. And we have also studied type of germination: epigeal germination and hypogeal. So here two concepts have been asked in a single question. So you have to match like that. So you know that castor and beans, these are the examples of epigeal or epigeous seed germination, whereas gram and maize show hypogeal, because maize is cereals. All cereals are mostly hypogeal. Gram is hypogeal. Okay, so you know castor, which shows epigeal germination. Castor is endospermic or albuminous. Okay, albuminous and epigeal. Beans are epigeal and ex-albuminous. Similarly, gram is hypogeal and it is ex-albuminous, and maize is a monocot which is showing hypogeal germination, and it is albuminous because it consists of endosperm. So after proper matching, you will get the answer. Option A is the correct answer. An ex-albuminous seed with hypogeal or hypogeous type of seed germination is castor, cotton, sunflower, gram. Here the answer is gram, because castor, cotton, sunflower, these are albuminous seeds, endospermic seeds. So D is the answer here, which is an ex-albuminous seed, as well as it shows hypogeal germination. D. The seed vigor index of a seed lot, not a single seed, but many seeds in a single lot. The seed vigor index of a seed lot is calculated by the formula given by scientist Abdul-Baki and Anderson in 1973. So this formula is, so this index is calculated by adding percentage germination, percentage of seed germination, percentage seed germination and seedling length in centimeter or millimeter, subtracting this from this, or multiplying this from this, multiplying this with seedling length, or dividing this with this. So what is the exact formula that was given by Abdul-Baki and Anderson in 1973 for calculating the seed vigor index of a seed lot? So here the answer is C. So when you multiply percentage germination with seedling length in centimeter, it will give the seed vigor index. This is the one vigor index given by the scientist. Another vigor index we called as vigor index 2 is also there, in which case there is multiplication of percentage germination of seeds with the dry mass of the seedlings. Dry mass of seedling is multiplied with percentage germination. So both of these formulae are used to calculate the seed vigor index of a seed lot. So here the answer is option C, that is the right answer. So again, these same references, seed dormancy and seed germination, many references you can see here. So of course, I know you must have subscribed up till now. If not still, then you can subscribe, bell the icon button, then you will get notification. Thank you. Thank you very much for kindly listening to this video also. We'll meet again with some new topics from this portion, plant physiology. So up till then, bye. Thank you.